Welcome to solverfy.
Hi everyone, my name is Eric and let's answer problem 51. So in this question, there are three parts to it and the focus of this is on the concept of gel electrophoresis. Now gel electrophoresis is a laboratory technique and it's used to separate molecules based on size and charge. So these two principles make up gel electrophoresis. Okay, so knowing that, let's look at each part. Part A is focusing on amino acids. So remember, amino acids are the molecules that can come together and build proteins. So amino acids, we're given three amino acids in this question. We have alanine, we have serine, and we have histidine. Now, from these three amino acids, the clear difference is they have a different size, right? So that is a part that we can separate these amino acids from. And for charge, we know that from the beginning of the question, they have introduced that the samples will be placed in a solution of constant pH. So charge will most likely not play a definitive factor. So our focus will be on the size of the molecule. And we can see that visually, alanine is the smallest because it has the least complex of the chemical structures, and the histidine is the largest, especially since that it has that extra ring. It gives that molecule a larger size. So when we are looking at a gel, and we know that it moves from a negative to a positive terminal in a direction where the electrical current will take the molecules from one side to the other. And we have starting wells where the samples will be placed in, and it will move accordingly with the electrical current. And we know that from the beginning, towards the beginning of the gel, our molecules will be large in size because the larger your molecule is, it will not be able to travel further down the gel. It gets trapped in the pores of the gel. And as we move away from the starting point, our size will be smaller. So, taking that into account, we know that histidine as the largest will be towards the beginning. And then as we progress through the gel, we will eventually run into where the serine would be located. And finally, our alanine as the smallest molecule will be towards the end of the gel. So this is how the amino acids will separate. And of course, alanine will move furthest down the gel because of its size. So that is part A. Now, part B, we're looking at a different scenario where we are introduced about a new protocol. And our objective is to analyze the gel, right? So by reading the scenario after taking into the information, we know that there are two lanes that are being presented to us, okay? So the first lane with one band is the sequence that is being inserted into the plasmid, okay? Then the next part of the gel is the second lane. So this second lane has multiple bands everywhere. And the direction of development, it says that it's moving from right to left. And the second part, the second lane is the lysate. So the lysate is what's being broken up and then it's going to be digested and then it's fragmented out so that it can be displayed in total. So this lysate is actually the plasmid. So plasmid is a circular piece of DNA and it will be broken up into different segments and then those segments get run onto the gel. Okay? And the question is whether the protocol was successful. So after looking at the gel, we can tell that it was successful. So yes, successful. because of this area right here, where the sequence matches the lysate. Because that the sequences are located in the same area, in the same size, it shows that the lysate contains the sequence being inserted into the plasmid, right? Because this sequence is most likely to be matching with the sequence that is being inserted into, right? So this sequence here would match this sequence here. And because they are located in the same region, we can tell that our sequence has been successfully uploaded and uptaken into that plasmid. So that is one way that we can verify that our protocol was a success, because the developed gel shows the two bands in the same area. And part C is asking for a way to determine whether the new DNA has actually been incorporated into the DNA of the host organism. And a way to do that, there are many different ways of evaluating that, but the simple answer is probing. So probing is a way to actually identify whether or not that sequence has been uptaken into the DNA. And one kind of probing is DNA probing. So the DNA probe would actually hybridize to whatever sequence that we're looking for. And it would be a way of identifying. So let's say that this red strand is our DNA that we're looking for. So this is our sequence of interest. If we had a probe, that probe would be complementary to the sequence, and it would bind and hybridize. And this probe would contain a radioactive molecule. So this radioactive molecule would be a marker. So this marker would actually do something like a fluoresce, or it would be even like a light up sequence, and you can see visually whether or not that DNA has actually been taken up or not, right? So all of the hosts that have the fluorescent marker, we would be able to tell that, hey, our sequence is inside the plasmid, so therefore our DNA has been uptaken successfully. And so after looking at all of these questions and reviewing all of the steps, I hope this makes things a little bit more clear for you in understanding how the gel electrophoresis works and how we can read it and use it to our advantage. Thanks for watching, and I'll see you next time.